Hey everybody, today on the Demystify Sci podcast, we are meeting up with the science detective, Elisabeth Bik. She's had a fascinating career, starting off in microbiological research, but she had some experiences along the way concerning various minor infractions that she detected going on in the laboratory and with regard to her own research being plagiarized in various ways. And she started looking into it and found that malfeasance wasn't so much the exception but seem to be the rule in basic institutional research. Now, that's not to say that everything is garbage, but it is to say that there's a lot of garbage out there and a lot of really pernicious gray zones and negative incentives that seem to be pushing people at all levels towards putting out results that don't hold up in the long run or things that reinforce paradigms that don't need to be reinforced. And so we explore all these issues with her today. It was a really, really interesting conversation.
Yeah, I've been following Elisabeth's work for many years now because I came across an article about this woman who was able to, by eye, identify where people are manipulating images. And so what happens is that people will reuse the same image across different papers. Sometimes they'll take pieces of an image and they'll rotate them and use them in a different figure in the same paper. And all of this comes down to the fact that people are faking data. And normally you expect to hear that somebody who's been responsible for more than 2,000 retractions is using some kind of advanced computational method for being able to identify these things, but Elisabeth's path into this was just identifying weird stuff by eye. And that just, it was that's a person that we have to talk to because it's just such an unusual and really world-changing skill because everybody knows that there's problems in the sciences, and somebody who devotes their time to cataloging them so clearly is really doing a huge service for the community. And so our conversation is super interesting, covers everything that Shiloh mentioned, and we kind of land on what I hope will be the topic for the next conversation, which is, what does it really mean to do good science in the first place? We say that science is the search for truth, but what does that mean?
What does it mean inside of the context of an institution too that's reinforcing old ways of thinking fundamentally?
Yeah, absolutely. So I hope you enjoy the conversation. For all of you that enjoy our conversations and are supporting us already at Patreon, I want to say a huge thank you because there's no way that we could do this project without you. We actually talked to Elisabeth that she uses the same format where she's supported by patrons, and it is one of the most liberating and satisfying things that I have ever done in my entire life, which is that I feel like I'm doing a service for people, and there is a great resonance between us and the viewers, and the patron is kind of the place where that resonance comes into the fold.
Yeah, it's kind of like, speaking of incentive structure, it's kind of the most healthy incentive structure imaginable, where you guys who are enjoying these conversations that we are putting together are the ones making it happen financially as well. I think that's kind of the most farm to table imaginable structure, because we don't have to deal with advertisers. We don't have any institution breathing down our neck, corporations telling us how we should think about things, and we aim to keep it that way. So come on over to patreon.com, check it out. You can really just give as little money as you possibly can manage. It probably seems like nothing to you, but all those little donations add up to the point that we can spend all of our time working on this project and hopefully increase its power and impact.
If you can't do that right now, that is totally fine. You can leave a comment, you can review the podcast, you can follow us on Twitter, on Facebook, you can
come out to the conference. We're having our first live event this April.
That actually doesn't work if you don't have any money, because that's going to be quite expensive. But you should definitely still come out to the conference.
Maybe. One-time payment, and you get a chance to hang out, make some friends, meet some of the speakers, some of our guests that we've had on the show to this point. And I'd love to see you there.
And for now, enjoy the conversation with Elisabeth Bik, and we will see you next time.
The scientific revolution starts now.
The Demystify Sci podcast.
Let us see in the dark.
I'm Elisabeth Bik. I did my PhD in the Netherlands, and I moved to the United States about 20 years ago. and worked 15 years at Stanford on the microbiome of humans and dolphins. And at some point during that time, I found out that somebody had plagiarized one of my articles. Not the whole thing, but they had stolen a couple of sentences. And that made me very mad because somebody had stolen my sentence. And this was an article that also contained sentences from other people. So these authors had stolen sentences from a bunch of people and put them together in this new review paper. And yeah, it just made me mad. And
How did you figure that out? I'm sorry to interrupt you, but yeah, how did you notice that?
Because I heard about plagiarism and I thought, let's go on Google Scholar and type in a sentence I have written between quotes. So it was a short sentence, half of a sentence, something like that. And I could have picked any sentence from the review paper I had written, but I happened to choose one that was stolen by others. And they had stolen 2 short paragraphs of me. So
did they cite you?
I don't think so. There were 2 versions, actually. It becomes very complicated, but I believe they did not cite me.
Oh, wow.
Yeah. But since then, I worked on hundreds of these articles because if you search for a sentence, you might find just the hit with the article that it should be in, but sometimes you find other hits. And so it became this hairball of plagiarized sentences that I started working on. And this was all in my spare time, in the weekends, in the evenings. And I did that for several years. But in the meantime, I also found, by another coincidence, I found a PhD thesis that had plagiarized text, but it also had duplicated images. They were the same images. They were of protein blots, Western blots. And it was the same image, but it was flipped. It was mirrored. And I could recognize that image and remembered it because it had 4 stripes and a little dot. And because that dot was in the corner, because it was flipped, now it was in the opposite corner. And I recognized that too. And I was already angry with that PhD thesis because it had plagiarized text. But I also was now even more angry because they had apparently used the same photo twice to represent 2 different experiments. And that made me realize that I have some talent, I guess, for recognizing patterns, for recognizing photos, and even if they're rotated. And these PhD chapters had been published and peer-reviewed also. So it had gone through peer review and nobody noticed it.
Had you noticed that before in your life, that you had a really good memory for images?
Yes, because I always look at bathroom tiles and floor planks. If they're laminate floor, so not natural wood, then, or porcelain tiles, so not natural stone, then they will often have repetitive patterns. And I've always noticed that. You go in a restaurant, you go in a hotel, and you see these patterns in the carpets or in the floors or in the patterns of the tiles. And I've always noticed that. And I thought everybody saw that, but something you don't really talk about. But I guess I have that maybe a little bit more than other people. And now I found a reason to actually use that for good use, to use that to scan scientific articles for photos that have been reused.
Most people wouldn't do it as a part-time thing after work. And so what excited you so much about it that caused you to pour so much time into it? Was it just the sense of injustice that people were getting away with using ideas that weren't, or using text that wasn't theirs?
Yeah, and images that were perhaps representing other experiments. I guess it was a sense of injustice, a sense of this should not happen in science. And when an image is reused, sometimes it could be an honest error, but when it's rotated, mirrored, that seems to have been done on purpose. So that made me very angry because for me, I feel that science should be about finding the truth. And we can have a very long conversation, of course, about what the truth is. But I feel if you plagiarize and steal images, or if you reuse images to represent something else, that is a deviation from that truth. And so as a scientist, I just felt this urge that this should not be a scientific paper. This should be commented on and perhaps corrected or retracted.
Yeah, with the plagiarism thing, with people lifting text, first of all, like I've done this with our blogs and stuff, like people are completely repurposing writings and even like from my thesis in grad school too, like you can find that it's so common that I wonder if it's even possible to hold it back in some sense. And to that end, like especially with the future of, we have these artificial intelligences that are essentially just grabbing up people's ideas from all over the place and just slamming them together. I just wonder if that waterfall can be held back. But it seems like a very different problem than manipulating an image to prove a point, which is like, there's something very corrupting about that threatens the very heart of what we're trying to do in studying nature, right? It's almost like it's a real force that's working against you in a way that lifting text seems somehow less threatening to me. What do you think?
Yeah, I agree. And that is the reason why at some point I switched from working on plagiarism to working on these images. Because like you said, I felt if you plagiarize or copy text, it's bad for the scientist whose work has been copied, obviously. It's not good. And it's
kind of bad for you too, right? Like,
yeah,
you're not digesting anything.
Exactly. And it's you're not showing me that you're a scientist who has given this topic some thought and has some of their own ideas about it. But in a way you could argue it's not bad for science because it's just repeating what somebody else has already said. It's not creating a false narrative necessarily. So when I found these images that had been duplicated, I thought that was even worse because now one of these two experiments, represented by the same photo, one of these two didn't happen. And I don't know which one, but maybe, they didn't like the results. The authors thought that it should have looked a certain way and it didn't. So they just took another photo and represented that as their experiment. Now that is lying. That is fabrication or falsification. And that is another, in my opinion, much worse form of misconduct than plagiarism.
I mean, it seems really criminal on the way that it's presented like this, but I think that it's worth putting ourselves in the mind of somebody who's doing this for a second, because I don't know the specifics of this Western blot that you're talking about, but it's very possible that they had an image that still represented the same, like it looked basically the same, but it was just a crummier exposure. It just looked bad for some reason, and they figured, well, I'll just, the conclusion is still the same. The data is still the same-ish. I'm just going to show this one because it looks nicer. And right, so that's like, I think that's a lot more understandable for the average one of us who's listening right now.
Is it though?
A little bit. It's at least you can see the small line that you have to cross, right? Because in your mind, you're forgiving yourself because you're like, well, I'm still representing the truth. I'm just doing it like in a more aesthetically pleasing way. And so, I assume once you cross that line, then it's like maybe you're starting to think, well, what if I just, don't even do the experiment, right? And like it's a slippery slope, but I think that it probably starts off with something sort of benign and like
the gateway drug of data manipulation.
Yeah, that's absolutely right. I think a lot of these cases start perhaps in a very innocent way because it's very easy to cheat in science that in ways that do not leave any traces, like. And we all present our best experiments. This is very natural, and it is indeed a slippery slope. It's often the lines between what is okay and what is not okay anymore is not as clear as you might think. You might, for example, if you try to get some assay working, some PCR, or you try to grow the cells or your bacteria, you might try many different things. You try adding this, a little bit less of that, you raise the temperature, you change the pH, whatever, and suddenly now it works. And so you only present typically the result that worked. And all the other attempts that you did that failed, now you know, okay, well, that didn't work because the cells didn't like that pH, or the enzyme didn't work under these salt conditions or so. So once you figure out the best conditions, you present those data, and you sort of summarize perhaps in one sentence, like, we tried different conditions, but this one was the only one that worked. So it's very normal to leave out the results that didn't work to some extent, but it is indeed a slippery slope to, if you do now the experiment 10 times and it only worked one time under these same conditions, and you only present it one time, well, is that cheating? Because you know the other 9 times it didn't work, even though the conditions were exactly the same. So that is not good. And if you have a blot that had a stain on it, or a crack, or a hair or something, and you use another photo, maybe that is not as bad, because if you could show me the original photo with the hair or the crack, then I might say, yeah, that looks pretty similar. So you're right to have taken, well, not completely right, but at least you're not messing with the results. But the problem is we do not know by looking at a scientific paper what the original was. I can see that that these two photos are the same, but I don't know if the original looked very different or if it looked very similar, and that is the problem. So when I raise these concerns, I would love to be proven wrong by the author showing me the original, but many times, most of the times, they don't reply and they don't show me that original.
It's really, I was going to say, it's an interesting tip of the iceberg in some ways, because how many of these are Western blots? Like, what percentage of the misconduct is Western blots?
Ooh, I would say about 30-40%. There's also photos of microscopy photos, or photos of mice or tumors, or leaves of plants or so. So it could be whatever is in a photo in a scientific paper.
So if you can detect misconduct in the copying and manipulation of photos, there's a bunch of misconduct that is inaccessible to you, which is stuff that's actually numbers based. Because all you see at the end of the day is the chart. Most journals don't have you publish your full raw data. And so...
Well, they do, but it's not standardized in some format, so it's easy to, like, I mean...
the raw data file is somewhere maybe, but it's not like it's on the same place as the supplemental figures file is on a journal page. Like, you go read a paper on Nature or something, and there's not a button that's in the same place as supplementary information, download the raw data for most papers.
I think there is, but you can get to it off the main page or off the actual article. But the problem is that analyzing that data, I didn't personally do it, but I had a collaborator on my grad school project who helped me build essentially a whole analysis database to deal with the output, because the experiment I was running wasn't something that anybody else had a tool for. And so I just, we submitted all the raw data and everything. I'm just thinking, who in the world is going to go to the length to build a software to tear this thing apart? And I'm graduating, right? So I'm just kind of putting my data together and just handing it off to the next watch, essentially. And it's I don't know. We thought a lot about how to make that as accessible as possible, but at the end of the day, you can give them the code to analyze it and everything, but they still have to kind of build it out in whatever analysis software they have. And it's just there's no standardization, which I think makes it very difficult. And who's going to have the time anyways to do what you're doing, to go back and do something as tedious as crunching numbers all over again and creating curve fits and reestablishing these conclusions. And then how are you even going to argue that? You'd have to write an entire paper reanalyzing it. Just seems much more difficult with this quantitative stuff.
And so the question that I get from that is, what percentage of misconduct do you think is being detected by you and people like you?
Probably the tip of the iceberg. I have no idea. I mean, it's hard. So I did a check of 20,000 papers and looking at duplicated images within those papers. And we found 4% of those 20,000 to contain duplicated images. And about half of those, so 2% of the set, we estimated that that was done deliberately. For example, if an image was rotated, flipped, mirrored, stretched, or if an image had duplications within the photo, that is almost always done intentionally. So our estimate first was, okay, 2% of papers have these visible problems in the images, in the photos. But you're right, there's a lot of ways you can manipulate your data or change them that would not leave any traces. They could be in the numbers, in the line graph, somewhere in the originals, and it could just be loading something different and taking a photo and say that you loaded A, while instead you loaded B on your blot or on your microscopy slide. So there's many ways of cheating that would not leave traces. So instead of 2% misconduct, I don't know, maybe 10%, and but there's many ways you can cheat, and they would not leave traces. So it's just impossible to know. But it's much higher. It has to be much higher than 2%.
Yeah, some of the, I've seen a lot of things. So like I've worked in academic research for probably 10, 15 years. And I feel like I saw everything, like absolutely everything happen, and a lot of it going unpunished to varying degrees because it's so embarrassing. People don't want to make a big deal of it. I mean, I've seen people. One of the first ones I saw was actually really fascinating, kind of similar to you. One of the first big labs I worked in was this huge cancer lab at Ohio State, and when I was a technician there, and we had these huge lab meetings, like 50, 60, sometimes 100 people, a huge lab, just like whole tiers of hierarchical organization, dozen postdocs and research scientists, and huge. And the only time you'd really get FaceTime with the Uber PI was at these big lab meetings, basically. But he was really brilliant, and he had I don't know. He was an amazing person to have worked under, and I learned a lot from him. One of the things that happened at one of these lab meetings was one of the brand new postdocs was presenting for the first time, and she was presenting flow cytometry data, which is basically this like shotgun burst of dots on a page. And she presented for most of an hour, and then at the very end of it, he was like, "Can you go back to slide 3 for me really quick?" And he looked at it. It was one of these flow cytometry shotguns. And then he was like, "Can you go to slide 56?" And she goes to it. And it's like, and he doesn't say anything. But after the meeting ends, word slowly spreads that this postdoc is gone from the building, like completely been fired, let go from the lab. And it turns out that our PI at the time, I'm not going to name him just to be nice to everybody, but he had just somehow photographically known that she'd used the same control slide. And it turned out it wasn't just an accident. She had been doing this. He went back through her data in her office after the meeting, and it was a thing. And I'm just like, I was completely blown away that someone could remember a shotgun burst like that across the pages. So I feel like there's just a special talent that people like you have for recognizing these things that nobody else would know. But it was also the first time where I was like, wow, this happens? Like, people are doing this? And it just got worse and worse from there. I've seen people kill mice to make a drug look like it's working better than it is working. Like, really, really bad stuff that you'd never see a trace of unless you caught the person red-handed. So I guess it's just people have this idealized Baconian idea of how science works. You set up an experiment, publish the results, and it's just that if we're going to be honest about the scientific method, it's really subject to these institutional pressures, and that can't escape the discussion of what the scientific method is at this point, because are we actually getting this idealized truth out of all of these studies, or is there something else going on here too that we need to see when we look at these headlines?
Yeah, it's I think one thing to realize is that it's very hard to publish negative science. And so what we see being published is positive science that works, a drug that works, an assay that works, or the enzyme that does what the PI thought it would do. But behind every paper that gets published with these positive results, there's many that are negative results. Like I mentioned earlier, you try to make an essay work, but it doesn't, and you try different techniques. So all of that doesn't get published, but also a lot of things that do not work or are not reproducible, those don't get published. So there's this huge amount of data that never gets published because journals like to show novel and exciting results, and negative or reproducibility studies are a little bit more boring, perhaps. And so what you see is things that all seem to work. And if you then do a meta-analysis of a drug, then that means that all that data is so biased already that it looks like the drug will work, but all those hundreds of studies that might have been done and never published are not going to be included in that study. So it just makes science much more biased and harder to interpret.
What's the distribution of malfeasance and impact? Because there's a part of me that's like, okay, most papers don't get cited. You publish something and you need to publish it because it's your position at a university or some tenure necessity. And it just disappears into the ether. It's a paper that no one's ever going to look at again. And
I think most academic papers are have have been read 3 times essentially.
Yeah, exactly. It's crazy low. And so if there's malfeasance in there and that's where the majority of it is, I'm kind of like, it's bad and it's noise and it should be stopped. But then there's the other part of me that's like, okay, well, if the higher impact stuff is disproportionately immune to this kind of thing, then maybe we don't really have a problem. And so I wonder, through your work, have you been able to see if there's trends between...
Can I just attack on one thing to that? So we had this guest on the show named Adam Mastroianni. He's at Columbia too. And he wrote a really interesting blog on his Substack about this where...
The strong link problem, right?
Yeah, go ahead.
No.
I mean, his basic idea was it doesn't matter. So if there's all this garbage science being put out, well, people won't be able to reproduce it. It won't get cited. And therefore, who cares? Quit trying to police this stuff. Focus on the strong link, the studies that actually do break boundaries, and everybody knows that they're true because they try them out and they're true. And so why should we waste all this time even with peer review? Why should we even mess with it? Who cares about all of these crappy studies that nobody's citing anyways? And I'm curious how you would respond to that.
Are they mostly crappy studies that nobody's citing, or is that totally not the case?
Yeah. Well, it is. It is true that most of the papers that we find problems in, and I'm not alone. There's a whole army of people doing what I do. Most of these are low impact journals, cited 2 times, 20 times, not very high. But there's definitely a bunch of articles that are highly cited. One particular article that I worked on with a team, we found problems in, that had been cited around 2,000 times. And it was actually one of the articles that made a start for the beta amyloid hypothesis in Alzheimer's. And this is not my particular field, so I don't know all the details, but it was one of the articles that many other people had cited, was cited 2,000 times, and it was a lot of people reported after we found problems. Oh yeah, we could never reproduce it. But the problem is that those we could never reproduce it studies never appear in the literature. So people just see this paper and they think it's highly cited, and so it must be true. But it doesn't necessarily mean it's reproducible. And so many others found that they couldn't reproduce that study. And but you would never know that by looking at the citation number. So citation numbers is not necessarily a sign that a paper is good and that it should not have any problems.
Hey folks, quick reminder, this podcast is entirely listener supported and we want to keep it that way. So please come over to patreon.com, give the tiniest amount that you can possibly afford, and it will add up and make this project more powerful in the future and allow us to put more time into it. So thank you. Check out patreon.com/demystifysci, and let's get back to the conversation.
But I did find in my initial investigation of 20,000 papers that I didn't look at citations, but I looked at impact factor of journals. So highly cited journals like Science and Nature with high impact factors had fewer of those problems than low impact journals that are in general less cited. So I did find the inverse correlation. So if the impact factor was high, the chance of that paper having a problem was lower than a low impact factor journal.
Do you think that's because they're better at cheating?
Yes, perhaps. You nailed it. Well, it's hard to know. There's many ways of cheating. And maybe those people who publish in Nature and Science are, in general, they're very experienced researchers. They already have built up a name. And so it becomes easier once you have one Nature publication, it becomes easier to have another one. Whether that is correct or right or not, that's another question. But so those people might just be more experienced and might be better at hiding if there is something behind the scenes going on that is not correct, that is perhaps fraudulent. But it might also be that there's better peer reviewers, so these things get perhaps caught already during peer review. And while the lower impact journals might not have an editorial team looking at papers and scanning them for all kinds of problems like perhaps Nature and Science have.
Can you respond more to the danger of these low impact journals being full of garbage? Like, is that a legitimate threat to science, or is it just kind of an annoyance that nobody will really be bothered by?
It's hard to know because I can see it from both ways, but I feel those are polluting science. So they might still lead other people to follow particular leads, especially if it's a completely fake paper. There's this problem of paper mills that produce scientific papers that are completely fake, and they sell the authorships. And so those papers are probably nothing in that paper is true. It's all made up, and that would pollute science because you could argue, well, it's a low impact journal, it's not really cited a lot. But if you do a literature search, or if you're a grad student trying to find a new project to work on, you might see this paper and you might base your research on it. And of course, if it doesn't work, you don't cite it, perhaps. But it's polluting science. So I feel we should still worry about those things. And it's a sign that perhaps the whole peer review system is not working very well, and that there's many people who just publish something because they need a paper to publish. They don't care about the science, but they have this requirement to publish.
There's the whole behind the scenes aspect of it too that just occurred to me, which is that these people, their paper might not have a big impact, but they're still out there in the fields teaching students and mentoring a whole new generation, right? Even at these smaller institutions, maybe or I don't know where these low impact papers are coming from, but it seems like it's still in some source reinforcing an ethic that is, well, not reinforcing the ethics that students need to be appraised of early in their career.
Not only that, because I think that there is like a huge ethical problem that comes as the product of being educated by somebody who's like, this is fine. Like, this is just how we do things. We live in a culture where when people want to evaluate whether or not to do something or to take some kind of intervention or how to structure their diet or even things as basic as, I don't know, the best way to start your morning. Everybody wants a set of scientific papers to point to that tell them what is the best thing. And so everybody points to science papers as evidence in constructing the way that they live their lives. And so if we have this huge sea of crummy papers that you can point to justify any decision, and it's literally just based in lies, I think that's pretty damning. Even without considering what Shiloh's saying, which is this accumulation of just bad practices in dynasties of teacher and student.
Mm-hmm. Yeah, and it's easy to find any paper proving anything, like is turmeric good for you or not? And you can find hundreds of papers that will say one thing and hundreds of papers that will say the other thing, and which one is true. And I think that is because a science paper is not set in stone. It might be peer reviewed and might even contain the truth. It might not have been based on fraudulent data. But science is often a very hard process. I don't see science as like a set in stone type of sets of truths. We're all finding the truth. We all want to do that, but it's a process. We sometimes, for example, you might take a bunch of human subjects, and you might, convince them to enroll in your study, and you do that, and something works, the drug works, or it doesn't work, and another person sort of does the same thing and finds the complete opposite. And why is that? Because that sounds, I think, for the general audience, very conflicting. You would expect that if you would reproduce a study, it would be the same. But perhaps you are studying people in a different country who are following a different diet, or perhaps people study a different set of mice, a different strain, or to work in female mice and then repeat it in male, and it might turn out very different. And it's very often these little these things that are these details that might make a lot of difference in the outcome of a study. Maybe somebody injects the drug, and the other study looks at it ingested by mouth. Those things could all make a huge difference, even though we might all conclude that this drug does work or it doesn't work. But it's so complex to look at all these details that for a general person who's not a scientist, it comes across as very confusing why all these studies point to different things. But science is a process, and we're all trying to find the truth, and it sometimes takes decades before we know how we should do a study and get the same outcome.
Yeah, and so you have to have a system that leaves space for that. And I think the argument is that the only way that you can possibly do that is by making sure that it isn't polluted by misconduct. Because if the goal is I think that you said something that's really, really key there, which is that sometimes it takes decades to get to the point where you're like, this experiment works when you do it like this because of this, 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 and this. And then people can reproduce it everywhere. And then all of a sudden, you've raised the likelihood that you're looking at a real phenomenon as opposed to something that's just kind of noisy and lost in the hands of the experimenter or the system. And even then you haven't fully proved it, because I think that there's also another huge layer to this terrible onion, which is the problems with animal models and the systems in which you're studying things. And so it's even if you do everything perfectly clean, then you still run up against this problem of how do you get out of the test tube and into the human or into the world?
Absolutely, yeah.
And so, can you steelman Mastriani's argument? Is he just kind of it'll sort itself out in due time?
Well, the other half of Mastriani's argument is that the peer review system does a lot of negative things too, because people who are at the top of some intellectual hierarchy have a great deal of interest in maintaining the predominance of their paradigm that they've been advocating for their entire lives. And so you don't really necessarily get this objective review of your theoretical concepts at the end of the day. You often get it in the context of what's popular of the day as well. And so it does, in his opinion, it does a lot of harm. And then the fact that people are, maybe publishing garbage is not a problem, as we've discussed, because it doesn't get cited and who cares. One thing that I wanted to kind of pivot to is that it's all well and good to punish people for crimes, for sure. That makes a lot of sense to me. Stealing's bad, right? I'm glad there's a law that somebody can't just kick in my door and take my computer away from me right now. That's really nice. But there's also this other aspect where you can imagine a point that stealing becomes sensible, rational, at least, because let's say in, the Eastern Front in World War II, where nobody has any food, and it's you break into some Nazi storehouse to steal some loaves of bread or something. That's really extreme, but I feel like in a starvation context where people are really hurting for, in this case, maybe just for survival in a career, that it starts to reflect upon how we've structured the way that people are getting fed, maybe as a result of the crimes that are being committed. And I guess another way to look at that would be like, was this a problem in 19th century science when it was like rich barons doing all of the work? I tend to doubt it. I tend to feel like this is a modern problem that arose in the 1950s when technical science became a career, when the state started funding basic research for the first time. And if that's the case, then what does a better version look like that doesn't incentivize people to be, well, yeah. Yeah. I'll let you respond.
Yeah,
Incentivize people to misbehave.
Yeah, like, or to not feel like they're starving, or is that a reasonable, is there some way to remove that incentive?
Yeah, we're currently focusing too much on metrics, as I usually call them. Like we want a grad student to only graduate if they publish 2 scientific papers in a journal that has at least an impact factor of X. And so if you focus on those numbers, which are easy to measure, as opposed to let, some rich kid in the, 100 years ago stumble around in a lab and just do whatever they want, that is much slower, but not really driven by an outcome that they need to have. If we make grad students only graduate if they follow these guidelines, if they reach these goals, then we are incentivizing them to cheat. Absolutely. And in some countries, they have taken that even farther. For example, in China, there's a rule that if you are a medical student and you want to graduate, you want to become a doctor, you are usually not that much interested in science. You are interested in helping patients and treating them. But these medical students need to publish 1 or 2 scientific papers in order to become a doctor. And this doesn't make much sense to me because, again, doing science, doing research is different than helping patients. There are people who love both, absolutely. But most medical doctors are not necessarily interested in doing science. So asking them to publish a scientific paper without giving them time off to do research, without them working in a facility that has research, research labs, that becomes an impossible task for them. So they will just see it as a hurdle that they need to take in order to graduate. And so they might see an advertisement online on a social media site offering them an authorship on a paper that they might suspect is completely fake, but they need that hurdle, they need that checkbox, they need that thing to get published. So they'll buy an authorship, and this is, I think, a similar situation. They need that for their career, and so they will think, okay, that justifies the means. I'm just going to buy this paper, this authorship, and now I can become a medical doctor and not care about science anymore and continue my career.
It's interesting because I can see the justification for putting that requirement into place in the first place. Because you're like, well, you're going to be a medical doctor, you're going to be reading a lot of primary literature, you should know how the sausage is made so that you have a sense of how to evaluate papers that you're looking at, right? Like there's a lot of good that could be seated behind it. You seem like you're kind of tentatively, maybe not.
Well, I'm not quite sure. I'm not a medical doctor, but I would. I don't think medical doctors in general read a lot of the latest scientific articles. They might
You'd
be
want
interested
them
a little
to,
bit.
though, ideally.
They definitely don't.
No, but you'd want them to. In an ideal
You want them
world.
to, sure, and they need to be able to understand it. But I don't think you that they have the time in their career to do actual research. And I think you can drive a car without knowing how the engine works. You could perhaps read a scientific paper and interpret that without actually spending half a year in the lab trying to write your own paper. And I do think that is something different. And I think it's absolutely good if doctors could do science and do research for a while and let them experience how hard it can be. But very often they don't have time in their schedule. If you have these impossible requirements, you let people work 80 hours in a medical ward and then also expect them to write a scientific paper in their 3 hours, which they have barely any of left, then I think it's an impossible requirement.
Yeah, I definitely agree. I guess
I worked in this neuroscience. There was a neurosurgery lab one time, and the neurosurgery residents had this requirement to publish something at some point. But it's insane because they're doing neurosurgery, and so they're like doing these 14, 15-hour surgeries. And I remember we had this one guy in our lab who would just come in. He was supposed to be doing his research block, but he would just come in and just like put his face down on his desk and just sleep. And like our supervisor, it was one of those mega labs too, and my immediate supervisor would kind of organize the rest of us and the grad students to kind of do this guy's experiments for him, basically, because he needed to have that paper, right, to graduate from his residency program. And it was just kind of normal. It was just like, yeah, like we just do all this guy's work, and I don't think I didn't end up on any of his papers, probably for the better. It is institutionalized even in America, I think.
we reminds me, we had a medical student in our lab, and it's very similar. We were all like pitching in to do her experiments, and one day I had made a beautiful blot for her, and it was drying on the bench, and she came in after, I'm not sure what she was doing, after her medical shift, and she put her bag on top of my blot.
Oh,
and
no.
I'm just looking like, what? This is an experiment. You cannot put your bag on it. And she had no idea. So yeah, she was, after that, she was banned from the lab forever. And we did experiments, but it is crazy that these folks end up on the papers while other people do the work. That is, it's not necessarily falsifying science or science misconduct, but it's wrong. And I just felt that person should not have been on that paper.
Okay, so it's easy to imagine removing some metric that's a bit perverse with respect to somebody's ultimate career objectives. But what about in actual institutional science, right? You need a way to evaluate and promote people.
Mm-hmm.
Can you imagine, or have you come across any potential indicators of progress that are divorced from publication record?
That's indicators of like having made scientific progress.
Yeah, like how do you evaluate somebody in the absence? I mean, this is honestly even something I teach. I just lecture at the university, and I'm always struggling with how to evaluate students because, you have to put a number down at the end of the day next to this person, and you have to find a way to evaluate them that's not just allowing them to check boxes in this really robotic way. And what have you come across in studying this? What kind of ideas have you had about ways that we could evaluate people that are metric somehow, but perhaps not tied to, putting this kind of perverse carrot in front of their nose to just get it out there no matter what?
It's a tough question because metrics like how many papers did you publish and how many were in Science or Nature, those are easy metrics to focus on. They're very easy for, let's say, an institution who wants to evaluate 100 candidates for this one position of a tenure track professor. And they get like 100 applicants or maybe 200. I don't know. I think those positions are scarce. And so they would get a lot of applicants. And so it's easy to look at resumes and just score how many papers they have published and how many times those were cited and what their impact factors were. But they don't necessarily make a researcher a better researcher. It's sort of a lazy way of evaluating. But of course, then the question is, how would you evaluate better? And the answer is that is very, very hard. It's very hard to know if a person who has published 10 papers in middle and low impact factor journal, are they a better scientist than the one who has 10 Nature journals or science journals? It's so hard to know, and whatever you can think of will take much more time. And if you have that stack of 200 applicants, you don't have that time to evaluate all of them by some other metric. So I can see how it is an easy metric to look at. But I think perhaps getting rid of things like impact factor and only look at citations might be a good way that has been suggested. But I think having the ability to publish negative results would open up a lot of good researchers to also have publications on their list as a reward for their effort. You can think of a grad student who was fantastic, who did everything right, worked hard, asked all the smart questions, but now their results didn't work. Their experiments didn't work, but they cannot publish it because it's negative results. So I think that denies a category of smart, young, early career people access to become teachers and group leaders in science, even though they're really smart and good, but they don't have those papers to put on their resume. So things I was thinking about is that we should have ways of being able to publish negative results. We should have many more ways of doing that, and perhaps we can think about senior people starting new journals that will be focusing on negative results or reproducibility results. We can think about grad students in their first year of their PhD to give them a paper that is seminal in their field and try to reproduce it, and have that be rewarded either in terms of a publication or in terms of effort, and being able to put that on your resume. So not only put the papers you have written on your resume, but the papers that you were able to reproduce, but also provide a platform for that. You can think about journals perhaps being published by universities, like the Stanford Journal of Negative Results or something like that. I think that would be a fantastic way to give graduate students a platform to publish their results in, even if they didn't work, even if the outcome was negative, and focus more on reproducibility. So I hope we can focus more on that, because that will slow down science, obviously, if we're not all working on novel things, but it will in the end make science a little bit better, because I think there's a lot of hidden efforts that we will never see published nowadays, of papers that were not reproducible. And I think we owe it to the taxpayers' money that we can tell them if a result did not work, and we need to publish that.
I really love that.
You can imagine it kind of snowballing if you had a paper about reproducibility in the Journal of Negative Results because you couldn't reproduce the paper. And then it's like, then maybe that gets flagged so that other people try to reproduce it. And then you actually get to a point where like 10 different labs can't reproduce it now. And then that's actually a pretty cool project, maybe that has some visibility and impact all of a sudden.
And it also leads you to the next thing, which is, okay, well, why can't we reproduce it? Because some stuff is just inherent malfeasance, and obviously that's why. But I think that there's also a lot of these problems with techniques and models that right now are hidden, because I haven't seen a ton of work about reproducibility that's trying to figure out why something isn't reproducible once you get past the obvious issues, like somebody faked data.
Yeah, and there's, I've worked in a molecular biology lab, so I only have some insights on those. But for example, we had one particular technique where we did Southern blotting, like making DNA bands visible. And we found out at some point that if you were wearing gloves that were powdered, the whole technique didn't work. Your blot would be completely blank. And so there was an easy fix. You just worked with gloves that were not powdered. Back in the days, I think now none of the gloves are powdered, but back in the days we had powdered gloves. They were a little bit more comfortable. You don't sweat in them. And so a lot of people were
But they give you
that.
Cancer from the asbestos and so to wash.
Exactly. So this was before all those insights. But I'm just giving it as an example of like these little things that you would not even include in your methods. Those might make a huge difference. And I have another example where we did microarrays. Again, sort of an older technique. I'm dating myself here, but this was, and we worked in a lab where they said the ozone concentration in the air is really important. If the ozone concentration is too high, again, your whole microarray, there's no signal or very low signal. And how would you know if the ozone concentration is high? That's not something people would generally measure. If you work in a lab and there's like the lab printer is chunking out, big amounts of papers for somebody who's printing out a textbook, the ozone concentration because of the printer might be super high in that lab. But you would never know it that your technique doesn't work because of that printer. You would never think about that. So it could be very little things, again, that are not included in a method. You wouldn't say, okay, we did this in a lab where there was no printer or no copy machine or so. You wouldn't say that, right? And you wouldn't think that would make such a huge difference, but it could. And in some of these molecular biology technique, the way a shaker shakes, is it rotating or shaking back and forth? That could make a huge impact. And again, that is, you would just say it shakes at 300 RPM or whatever, and you wouldn't say if the shaking was orbital or back and forth or so.
I think people did actually figure out that orbital and back and forth was important because I remember in all of the micro papers we would write that it was an orbital shaker specifically. Right. And I think that at some point somebody did figure that out, which is,
yes.
I'm like, that's good. But I think that this would open up science to a lot more different kinds of brains because I'm a person who's motivated by proving somebody wrong.
Mm-hmm.
I'm definitely less Shiloh. This is to
that's what I was
Shiloh's horror.
thinking is I could actually imagine a whole new level of science fame for being the first to discover that some fundamental finding, maybe it's not completely garbage, but it's just not what we thought, and we'd built all of our ideas on it, right? Some little reinterpretation of some data that was essentially reified into this concrete truth that everything was built upon. How that could be retooled, and you would actually, as a student or whoever, could actually be quite impactful in that finding. It could be a road to promotion in some sense.
That makes my brain light up so much more than being I'm gonna go find some new protein that does some new thing. I'm no. I'm gonna figure out how some machine is broken. Sign me up. And that was, I think, why I suffered so much in grad school, because I was look, in order for us to do these experiments, we have to jump through insane hoops, and there's all these things that change the outcome of the experiment. I think that all of those things that change the outcome of the experiment are far, far more interesting than the experiment itself. But there's no space for that.
Maybe you can share your exact experience too, because you had a really particular close call with all of this in some sense.
I did. So I spent the first 2-3 years of my PhD working on this really insane protein inside the membrane of the Pseudomonas bacterium. And it appeared to behave in a time-sensitive manner to the electrical conditions inside of the cell. And so I was doing these really arduous experiments where over the course of 16 hours, I would have to take samples every half hour and then do protein preps and Western blots to show how the modification of this protein was changing with time, and we thought that it was maybe a really important piece of the signaling system by which the bacteria was sensing its electrical conditions. And at some point, I realized it was an artifact of how long the samples would sit on ice. And I remember the meeting with my PI where he was
during the preparation, I'm trying to understand. This has been a while.
Yeah. So you take the samples, but you can't process all of them to get to the protein stage immediately. You would have to collect 15 samples and then do the processing of them. And then if you put them on ice, they're sitting at 4 degrees for different amounts of time. If you freeze them, then you have a freeze thaw. And so there's all of these different steps from going from bacteria to something that you can run on a Western blot. That there's places where the handling, and it literally, because it's a time course and it's sensing the electrical conditions, it's basically everything is moving all at once. And I remember when I realized that it was an artifact of...
And did you like process the controls earlier in the day or something? Is that why it didn't show
No,
up?
what it was is that the samples that, as the bacteria grow, they start to produce different compounds. And what I really ended up realizing was that as I lysed the bacteria in the presence of the other compounds that they made as they got older, it made the protein look like it was being degraded.
Mm.
Because I would like let them sit, for 15 minutes, and they were sitting in the presence of these other compounds, and that was what was degrading the protein. It wasn't that the protein was like changing its expression relative to like environmental conditions. It was just being broken to the point that it couldn't be detected by the assay. And so it looked like it was changing, but in reality it was just disappearing.
And you got kind of lucky because your PI was like pretty much like, okay.
Well, he was just like, is this an artifact? And there was a moment where I could have lied to him and he would have been happy, I think, to hear that it wasn't an artifact. Right? He wasn't, I think that it's devastating when you have a student who spent like 2 years working on something, 3 years working on something, and it turns out that it's just noise.
Mm-hmm.
And I remember the conversation, and I remember how just completely gracious he was about it.
Yeah, that's incredible, because I can imagine a different PI being like, no, it's fine, like, we should just submit this and just...
Like keep working on it, figure it out, applying pressure. And what I could tell that his heart was broken, and what he really wanted me to say was like, no, I can figure it out. But when I told him, I'm like, dude, this isn't real. He was like, okay, we gotta figure something else out.
Wow.
And so it was this moment of like things coming together where I could see the path branch somewhere else, and it didn't. And I deeply appreciate how much integrity he had, because
now you can sleep at night.
Yeah, now I can sleep at night.
You never published it, and it's but
how did remind me how I got onto this?
I just asked you to tell that story because I think it's a really interesting close call with a moment where like there's all these decisions being made and you can kind of go, it's just like, it's so easy to just, I think whenever I study crime in general, I really think it's important. This is just following from, who's this guy? Viktor Frankl, he's written this incredible book.
Man's Search for Meaning?
Man's Search for Meaning. And also like, Unit 731 and some of these other stories where they study these really, what we would consider very evil people, but it just turns out to be so banal. Like these people are just making these tiny little decisions that snowball. And it's just, I think it's really important for us to not look at people who commit ethical infractions as monsters, but as people just like us, and how we could also like fall prey to like these tiny little decisions that can potentially lead us off the rails. And like you're lucky that like you have the backbone not to have done that, but also that you were in a supportive environment where your PI didn't like throw you out of the lab for having studied an artifact for 3 years,
That he didn't throw me out of the lab at all.
And so you can imagine a totally different situation where it's much more intense, where the PI's like, all right, well, this, I don't know if I can keep funding you. Are you sure you can't fix this? And you're just like, oh God, like,
right.
That's where I
guess I can fix it somehow. Like, I don't know. I'll, whatever the tiniest version of that allows it to just go to publication is.
But to like wrap that back to the original departure point is that if you have journals that reward people for messing around inside the reproducibility of experiments and figuring out why something isn't reproducible, all of a sudden you have this path towards taking all of that work, putting it to good use, and then getting people to come up with better methods. Because like I asked you earlier if most of the problems were with Western blots, because I'm like, Western blots have to be some of the most perniciously difficult, easy to manipulate, frustrating
huge
assays. Huge time investment. Like they're just assays that beg to be messed around with.
Right.
And figuring out how to fix that and figuring out like, hey, it's Western blots are the problem. Like what can we do that isn't a Western blot that is as affordable as a Western blot that is less easy to screw with? Like that would be a really cool thing for people to figure out.
They have those speed Western blots. Have you guys seen those? They got these like right at the end of my tenure with this stuff.
Oh, the digital one that just create this like fake a fake horizontal gray line.
No, there was
Oh,
this
okay.
there was this box that actually would run the Western blot
magic
really
box.
fast, like high voltage transfer,
Right.
basically. It was
What could
it was kind of
possibly go
incredible.
wrong?
Yeah, well, but it would allow you to do more more replicates in the same amount of time, so that you might not be like incentivized to, oh well, this one's maybe okay, I'll just use it.
Well, speaking about Western blots, the one type of Western blots that I see the most duplications in is the loading control, where you first you test your protein of interest, and you have your different conditions, and in one condition you have lots of expression, and in another condition the expression of that protein goes down, so you have less amount of protein on that blot. But you always have to run, then you have to reprobe your blot for a loading control, like a protein that always you're suspecting is always expressed, always pretty much at the same amount, like a household protein, like usually it's like beta globin or beta actin. And so the blot gets reprobed to ensure that the amount of total protein on that blot is the same. But it's a boring control because it always looks the same. All the bands are of the same intensity. And so that is the blot that I see a lot of duplications in. People don't want to run this extra very boring control experiment. They just grab an older blot thinking, well, it's going to look so similar as in what it should have been. It saves me, 4 hours of this experiment, or I don't know how long it would take. But I can go home earlier, right? I just grab an old photo and I mirror it, and nobody will notice that. And of course, I will notice it, but they might get away with it for many years. And so you see, some labs have reused the same loading control for years. It's their favorite loading control. They always grab it they shift it around a little bit and they mirror it, but they never actually seem to have done the actual experiment.
Wow.
Hey, do you think that I could refill my coffee really quickly?
Of course.
Yeah, because I have a few more questions to wrap this down. But I just I want to caffeinate myself again if it's all the same to you.
No,
All right,
we need our caffeine.
There are only a couple of more weeks left before we have Demysticon 2024. We meet in Austin, Texas on April 7th and 8th for 2 days of talks, workshops, eclipse watching, and conversations with people that are interested in the same esoteric subjects that you are fascinated by. So Shiloh put me on the spot asking about my grad school research and what exactly went wrong. And I have not thought about this in like 5 years. And he kept asking me, he was like, I still don't understand. Like, how did you realize there was an artifact? And I was like, I remembered that the thing that I was seeing was a size change. And the way that I was detecting it was a Western blot, but it was against a tag that we had added to the protein. And for a long time, we were like, we need to get an antibody that's specific to the protein itself. And my PI was insistent. He was like, when you add a tag to a protein, sometimes they behave in weird ways. And so we need to see that this happens when we're looking for the protein itself rather than the tagged version. And it was that when we got the version of the antibody that was specific to the protein, the effect disappeared. And so it was something about the tag that was inducing the protein to behave differently. And so I had been studying the effect of the tag on the protein, not the protein itself.
Yeah.
Which is why I hate Western blots.
And a lot of people will say, why are you looking at Western blots? Because there's many better ways of looking at proteins. I'm not sure what they would be. But the reason is they're photos. And so, and this was before AI, generative AI, made its entrance to the world. If you see a photo, at least you know that the experiment happened.
Oh, that cracks open a whole can of worms.
It does. No.
Yeah, like I like you just go into ChatGPT or whatever, Midjourney or something, and you say, "Hey, I actually need a blot that shows this, this, and this."
I believe this is already happening, and we would not necessarily know it is because those photos, I'm sure you're familiar with how realistic these photos look like nowadays. They look so realistic that we could recognize a person on it. And, we could easily use that technique to make fake Western blots, fake photos of cells, and this is probably already happening. And I cannot use my magic eyes anymore to detect duplications or mirroring or any manipulations in these images, because they're unique, they look realistic, and we can easily be fooled by them.
I guess the plus side would be that the policing technologies would also be improving at the same steady rate. So you might be able to use some software that has a similar back end that's able to determine whether something comes from, it can maybe examine like the nuances of the light, or I don't know what, but could maybe keep up somehow.
Yeah, that is where I'm not a computer scientist. The computer scientists tell me, yeah, we can tell very often that these images are computer generated. And you can think about certain signatures that, I don't know, your lab microscope will leave on a photo to ensure that it really came from that microscope on that day. Some artificial handshake, or some computer handshake, or metadata that is associated with that file, which I'm sure you can also falsify if you wanted to. But there are probably ways. But what I'm doing, looking at photos, yeah, that no longer would work. It's very
Can you
worrisome.
tell us some horror stories of like the most egregious cases that you've seen?
Oh, well, many. I mean, usually it involves, the really bad ones involve photos that have been manipulated themselves. So within a photo, you see the same cell or group of cells duplicated a couple of times. Or I've seen some photos with histology, so photos of tissues stained with some particular protein. And if you look long enough, you just see that certain things have been copied within that photo. So only half of the photo is real, the other half is just copied versions of parts of the other half. So and in a way they're fun to work at. It's almost like finding an Easter egg. Once you see them, you're like, aha, I'm gonna spend half an hour of my time trying to find all these duplications, that even the software, I'm using software to help me find them. But in many cases, our eyes are still better. And so the software might flag a couple. And so I know once a photo has been digitally altered, you can actually spend some time on it and try to find more. And I play this game also on Twitter, X. I will post these things on X, and I will say, image forensics, that's the hashtag, and I post an image from a scientific paper, find the duplications, and it's amazing that in some cases people will find even more duplications. So it becomes sort of a game with obviously a very serious undertone.
Yeah, it's really interesting to me that you've managed to parlay this into a career that doesn't have like an institutional, like you're not institutionally funded, from what I understand. So
you're not institutionalized.
How have you done that? Yeah, you're not institutionalized.
I'm Patreon funded, so I'm crowdfunded.
Nice. Us too.
So that generates a basic income for me, because in the US you have to pay your health insurance and stuff like that, and your food and your rent and your car and your gas and whatever. So that helps me not have to worry about my income. It's not it's not as much as I used to make in the past, but it's enough to live by. And occasionally I will do some consulting work, and I travel to give talks. I get invited, so I ask for a honorarium, but very often the amount I get is not enough to cover even my basic travel. So I'm not really making a lot of money on those trips. But it also gives me a lot of freedom. When I was still employed, when I was still working at Stanford, during the day I was supposed to work, and I did, obviously, like I was paid to work on my project, and I did that, and I only had limited time to work on this project of science integrity, of looking at images. So that now that I don't have a daytime job, I can just focus on this the whole day. And I also don't have a boss who tells me, Elisabeth, calm down. That's, don't criticize that person because that's a best buddy of me. Can you move on to a different person? No, I want to have the complete freedom to criticize the papers that I want to criticize without other people telling me that I should or should not be doing that. So
you had a guy sue you, though.
I've been threatened to be sued. I have not been sued.
What did he threaten to sue you with? Defamation?
It was harassment. It wasn't defamation. It was harassment.
Yeah, telling.
It was it's a French professor, and I think it's still active. It's called a plainte, like a complaint in France. And I think it's sort of a police report that could lead to a lawsuit. So they have a very different legal system than in the US. But he filed me. He said I was harassing him because every time I criticized one of his papers, he would get an email. Is that harassment? I don't know. I get tons of emails per day that I think are harassment, but I wouldn't sue another person for that. And for blackmailing and extortion, which was based on a question that one of his professors working under him asked me. That professor asked me on Twitter saying, "Do you want to look at particular papers?" And I said, "Sure, if you pay me for that as a consultant." Now, I don't think that is blackmailing or harassing, but maybe their English is not very good. My French is not very good either.
What was the misconduct that you had found?
So this is a professor who has published 3,000 papers in his field in microbiology and infectious diseases, and it was a range of problems. So some of the papers had image problems where there's duplications within images. So those are the ones I found the first because that's what I do. But there were other papers that had problems with ethical approval, where the same ethical approval number to do research on human subjects, the same IRB number, was used multiple times to cover a range of different topics. And not only was that, but in France, there are certain rules where you have to ask for that approval at a certain institution, and they did not do that. They only gave themselves permission. Their own institution gave themselves permission to do the research, and they hadn't obtained approval from the official organization. So there were all kinds of problems. I can talk for hours, but it was a range of problems, and in the end, other people also joined to criticize their papers. So I think he has now around 300 of his 3,000 papers that concerns have been raised, and we post them on a website called Pubpeer, where we post our concerns. And so about 10% of his papers now has a concern raised publicly.
You know, Pubpeer. Oh, go ahead.
Like, how much of that is just? How can you actually publish 3,000 papers? I mean, that's
yes,
a paper a day for almost 10 years or something, right?
That is a good question. I mean, so he's retired now, this professor. I'm not going to name names. You can look him up. Easy to find. But he was the director of the institute where he worked. And so I think what happened is that pretty much every paper that left the institute, he put his name on. And whether or not he was really involved in that research, that seems impossible. Because, if you've done research yourself, I think it's realistic if you're a grad student to maybe publish one paper per, year. That would already be a lot. But if you are a professor and you have multiple people working under you, one of these people might publish a paper. So let's say you have a lab of 20 people, maybe you publish 10 to 20 papers a year. But yeah, to publish 3,000, it seems that he has been very generous in adding himself as an author on all of these papers, which by itself is also not a good practice, right?
But very, very common.
Very common, yes.
By the way, how long did it take you to get to the point where you felt comfortable leaving your job to do this full time?
It was about 4 or 5 years or so. 5 years, I guess.
And did you start the Patreon funding pretty early in that?
Yes, I think a couple of weeks later I set that up. So I quit my job, and the next day I set up a website and a blog post and Patreon soon after that, and I started looking at papers full time.
I mean, for me, this is like really inspirational because I think that we're in this, despite all of these problems and the hurdles that AI raises and so forth, it seems like we're in this really unique phase right now where people, especially investigators, journalists, are able to move away from these institutional like safety nets
Mhm.
more than ever in the last at least 50, 100 years or so, 75 years since institutional science has been a thing. It just seems like we're in this really exciting moment where people are finding ways to fund their own research outside of these institutions. It points to a potentially really beautiful future.
It could also set us up for all kinds of other problems, because there is some value in having an institution with, committees and people who will help you write grants or get the grants, or people who make sure you follow all the rules, and that there is a safety net around you where, let's say you work on a statistical problem, but you want to work together with other people. I think if you work in a bigger institution, there will be other people who can help you with that. If you are funded by a particular company or a rich donor, you might not have that safety net around you. I'm just thinking out loud. Like, I think there are advantages of working at an institution for sure, but I agree that it is really nice to have other ways of funding. Although I've been very frustrated with the lack of response from those funders when I found papers with problems. A lot of funders don't seem to care. You write them an email and they're like, "Oh, it bounces," or so, or you just never get an answer, and it's frustrating.
Like NIH type founder?
Yeah,
funders?
NIH.
That's
So the NIH has the Office of Research Integrity, and so any research that
you
has
can only
been-
reach them by phone between the hours of 2 a.m. and 5 a.m.
yeah, something.
once a
No,
week.
they have an email address, but now lately they have been actually sending me an email back saying, the basic courtesy of saying, "Okay, thank you, we got your email, we're going to look into that." But the first 5 or 6 years that I reported my concerns to ORI, they didn't even reply. There was just no email back acknowledging that I had gotten my email. It was basically, I was throwing it over the fence and I didn't even hear it land. And it was just very frustrating. Did they get my email or not? And you sent a reminder, and again, nothing happens. And so I've been saying this on Twitter, and since then they actually are responding. So, but I now ask in my email that I sent to publishers, can you please acknowledge receipt of this email? It seems very basic thing I shouldn't have to ask for, but now some of them are starting to do that.
I wonder how much of it is they don't have the power or authority within their
Yes.
bureaucratic hierarchy to actually, like, somebody else is dishing out the money at the end of the day. I imagine getting that money back is a whole other nightmare that probably like 2 people know how to even pull off in the entire organization. So I wonder how much of it is just they don't have the infrastructure to act upon these
Right.
leads.
Yeah, if it's a smaller funder, let's say some cancer research donor or so, I can see that. But when it's the Office of Research Integrity, which is a federally funded institution, you would hope for something slightly better. But they seem
But
to
like
be-
if they're in the basement of the NIH, right?
Yeah, probably. It's probably just one person or 2 persons. I think they're heavily understaffed for sure. But, just an automatic email reply doesn't cost much to set up. And that already would at least give me the reassurance that my email has reached something at that institution, has reached the basement. But yeah, it is. It's just frustrating, the lack of response. So
I
when
can
I
imagine
report-
a beautiful world too, where someone like you had a place at an institution as well. In that, like,
That would have to be the perfect institution. Like I'm just
but I'm
so
always trying to design the perfect.
I know, this is what I
love.
love. Because it's just like, look, the institution needs to be trustworthy
Mhm.
because it has tons of taxpayer dollars flowing through it. Somebody who's coming around who's like, hey, 10% of what you fund is garbage, or even 2% of what you
Or
fund
90
is
in the
garbage.
case of like mouse research.
Yeah, right? Because I mean, I think that if you bin that even further, it starts to turn crazy. But they have a really, really strong incentive to not pay attention to you until it is literally impossible for them not to. Because
Correct.
there's just so much weight behind the facade of no, everything is okay. We're spending money appropriately. All the checks and balances are in place that need to be in place. And it's just, I just don't know how else an institution could work. Like, what is an institution that is eternally content to say that it was wrong? Can an institution even look like that?
I think that would make an institution look much better and more honest and more caring about the integrity of research than an institution that is trying to hide these concerns and is trying to do all the investigations behind closed doors and never seem to come with an outcome. I think it makes a much better institution if you admit that there was an error and show the world that you care about it and that you do something about it. But unfortunately, most institutions are sort of playing this game where they drag and they postpone and it takes 3 years to investigate these cases. And if you read the timeline of what has happened, it's frustrating because they have guidelines, beautiful PDFs of hundreds of pages online where they say, okay, we do this phase in 6 weeks and that phase. There's usually an inquiry phase and an investigation phase, and these phases will take that long. But if you then see how they actually do these investigations, they take years, much longer than they say that they're going to do. And I think they just hope that the problem will go away. But I think with the power of social media, and social media has a lot of bad things, obviously too, but it has the ability to raise concerns in the public eye. And I think now institutions are starting to realize it's much better to do these investigations quicker and a little bit more in the open, and at least tell other researchers which papers we can trust and which ones we cannot trust, because it's not serving science if you do these investigations behind closed doors and never come with an outcome of which papers were found to contain misconduct.
Do you think that Pubpeer is playing an important role in changing the timeline of institutional response? Because this is actually a website that Shiloh and I had the idea of building, where it was like, wouldn't it be fantastic if there was like a Wikipedia type place where people could discuss the contents of papers and evaluate them and judge them, and you kind of like combine it with Reddit so that you could like upvote the most responsible posts. And I was really jazzed on it, and somebody was like, hey, have you looked at Pubpeer?
It already exists.
And it was like, oh.
Yeah, isn't that frustrating? Any idea you come up with already, somebody else has done it.
But actually, I really genuinely appreciated it because I was
Yes.
starting to think about how much work it would be to produce something like that.
Yeah.
And I was just so relieved to go out into the world and find that somebody had done it.
Yeah, and the reason is that it's not, at least for speaking for myself, what drives me, it's not really to bust people and make them be fired, get them fired because of misconduct. It's to warn other people that there might be a big problem with a paper. If you are a researcher and you base your hypothesis on this particular paper, but it has a problem, how can I share that? If I see that problem and other people have not seen it yet, how can I share with the world that this paper has a big problem and you should probably not base your entire career on this particular figure or experiment? So for me, Pubpeer fulfills that role. It's to warn other scientists. You need to have a plugin installed, and so it will alert you when you do a web search and you land on a particular paper's page that you can see a green banner and it has a Pubpeer command. And you can click on it and see what other people had to say about it. So we hope to fulfill that role of warning other people. And because there was no such thing, it was very hard if you had a concern about a paper. Some journals allow you to write in a comment, but very often these comments are strictly regulated, like they need to be a maximum of 250 words, or they need to be sent in a month after publication, and otherwise we don't accept any comments anymore, which is ridiculous, I think. It's very limiting. And so they make it very hard for other people to write concerns about these papers, and they might not even decide to publish it. So it's very frustrating. So I feel Pubpeer fulfills that role where people can raise their concerns in the public eye and invite the authors to actually come in and say something back, which very rarely happens. Show me that blot, show me the original and prove me wrong, or show me that you really obtained permission to do this ethical research, the ethical permit on these people, or that your tumors weren't as large as I think they were, that you were not maltreating your rats and your mice, and show me that I'm wrong. But very often the authors do not reply, which is also weird.
Do you believe in a path to redemption for people who have been caught red-handed doing these things? And like, what does that look like? Is there a Department of Corrections available for for bad researchers?
Oh, well, there's many scenarios. So I have certain scenarios that I think of when I think about what drives people to do misconduct. Where, we discussed earlier, sometimes the means might justify what we're doing wrong. Like if you really are forced in your career to publish a paper, you can sort of see that people will just buy an authorship. And there's other situations where I feel also in a way a little bit sympathetic for the person doing the misconduct. And one situation that I always think about is that of a person who has found a job as a postdoc or a grad student in the lab in a big name US university. But this person is on a visa. So if you are on a visa and you lose your sponsorship, if you're fired, and I've been in this situation myself coming from the Netherlands, you will immediately have to go home to your country. So imagine you're that early career researcher in the lab and the experiments don't go well and you don't have a nice supervisor who is sympathetic. Instead, you have a supervisor who is pushing you and who is bullying you and who's saying, I can hire another person who makes this experiment work. You're useless. And I'm going to, unless you give me these results by Friday, I'm going to find another person to do this. I'm going to take you off the topic and I'm going to let you go back to your home country. And I think in a situation like that would drive people to cheat because they feel they're in a horrible situation. There's almost no way out. So I think in these cases, who is responsible? Is it the person doing the photoshopping or the whatever misconduct they can think of, or is it the senior person who is responsible? And I'm not very tolerant for senior people who drive their researchers under them in their team to do misconduct. I have no sympathy for them, and I feel that they should leave academia. But very often it's the junior people thrown under the bus. And I feel sorry for them because they felt this was the only way out of the lab with a nice publication. And it's the senior people who get away with it. And there's no good system that will take these people, take their privileges away. In many cases, people with dozens of Pubpeer comments are still employed as full researchers, full professors, or vice deans of something. And there's just no punishment for them.
So I didn't follow very closely the Stanford story. Is this kind of something that happened at Stanford where there was knowledge about retracted papers and inconsistencies for a long time before consequences really started to pay out? Or was it something that all at once there was this slew of discoveries and the repercussions came very quickly?
Well, very often you hear stories where people said, oh yeah, we knew this was going on. Not specifically for the Stanford lab, perhaps, but very often people were not able to reproduce those experiments. People knew that the lab was led by a bully. But yeah, there was no real action. But suddenly, because of Pubpeer, because of student journalists, because of social media, suddenly these cases are brought together. Somebody writes a blog post that for some reason becomes viral. And I've been doing this work for many years. So in many cases, these stories are not picked up by the media, but sometimes they do. And then suddenly these cases are in the news.
What exactly happened at Stanford?
I'm not sure what led to it, but one of the Stanford Daily, which is a student-run journal. They got a tip about papers from the Stanford president had been flagged on Pubpeer already years ago, like in 2015 or so. And this student journalist, Theo Baker, wrote a piece about it. And he actually, before he wrote that piece, he asked me to look at these papers on Pubpeer and the concerns. And he's not a molecular biologist, so he needed some help in explaining if these things were very serious. And so I helped him a little bit with that. And I found some more problems also. So in the end, this article came online, and yeah, that was very explosive. The Stanford president was very angry that these allegations were done and wrote very stern letters to the students who wrote the article that their legal team should look into this or things like that, and he should take down some stuff, and that all these allegations were ridiculous. But there was some solid evidence that the student had obtained from people working at Genentech, where the Stanford president previously worked, that there were cases of misconduct and that papers should have been retracted, but that the president never did that. And so there were all these people who had testified the same thing. And unfortunately, in a later investigation, they withdrew their statements because they weren't offered complete anonymity. So they didn't want to proceed with their statements, but it seemed, at least from my point of view, that those testimonials were pretty serious and damning for the Stanford president. So in his previous roles, there were several occasions where misconduct had happened under his watch, not by himself, and so in the end he wasn't found guilty himself, but it happened multiple times in the lab that he was leading, that people did misconduct. And so you have to ask yourself, what was going on in those labs? Is it the personality of the PI that drives multiple people to do misconduct?
Well, and it's also, he was at Genentech when this was happening?
Yes. And before that, I think at UCSF, San Francisco.
Which are basically the same thing.
Yeah, I worked in Genentech College.
That's a different discussion. Yeah.
I'm just saying, we both worked there for a long time. And Genentech they had their fingers in the pie, let's put it that way.
Yeah.
But I'm like, there's so much money mixed up in this, right? Because this is, if I remember, I'm really embarrassing myself for not knowing the details of this. This was an Alzheimer's research project, right?
Yes.
Okay, so Genentech makes a drug that they're then going to get tons of money to do a clinical trial for, and they're going to give it to tons of people, and there's a huge financial incentive to produce research that gets you to the level of clinical trial.
Correct.
And the consequences of screwing around inside of that system are massive because you waste so much money that could actually go towards something that will help resolve what to me is maybe one of the most harrowing and horrifying diseases that is common in our world. Like, there's definitely worse stuff. Like, my grandma died of Alzheimer's. That was one of the darkest things I've ever seen in my entire life. And so the idea that there's this guy who's putting out research that is leading to just straight up false results, like, it feels so much more consequential than somebody who's puttering away inside of an academic research position. And it's like, yeah, you're going to get some grant money from the NIH or whatever, but it's not the same caliber of funding, the same magnitude of funding that you get for clinical research.
Yeah, and Alzheimer's is, like you said, it's a disease that affects all of us. We all have either family members who died with Alzheimer's or are currently suffering from the disease. It's devastating, and there's just no drug against it. And there's a lot of money being poured into research for obvious reasons, but it might just be that because there's a lot of Alzheimer's research going on, the percentage of misconduct, I'm not sure if it's higher than in other fields, but the absolute numbers are going to be staggering just because there's so many labs working on this disease. And there's many other cases, not just the Stanford president. It's been several other cases in the news where Alzheimer's research has been found in particular labs to contain digitally altered images. And it's devastating to think about it, that that money could have been spent on alternative routes, alternative studies.
Somebody on Twitter, I guess Eugene on Twitter, is curious if this reproducibility crisis scales back once you get to human patients. Is it more difficult to pass these things off as real once you get outside of the laboratory? Have you found anything like that? Or is it the opposite?
Well, for me to look at clinical trials is hard because they don't have images usually. No, they don't have photos, right? They just will have a table or some chart or so. But I do think that one of the reasons, and there's many, and I'm not sure, what a percentage is or anything, but one of the reasons that clinical trials might fail is that the research in the lab was changed a little bit, was tweaked to look better. And once you do a study in, especially in phase 3, where you have hundreds or thousands of patients involved, usually recruited at multiple sites, multiple hospitals, I think it's much harder to fake your data. It's easy to perhaps do it when you're still working in the lab and you want your results to look better, so you get a new grant and you get new investors to be interested in it. But once you really test your product in a group of 1,000 people with multiple labs involved, multiple hospitals, I think it's much harder to cheat, because now if you want to cheat, you would have to convince all these people to cheat. And some of them are not going to do that. And some of them are going to raise the alarm with the FDA or so. So I think it might be one of the reasons why some clinical trials will just not work, even though the lab results look amazing. And of course, there's many other reasons. For example, animal results would not translate to humans, or the labs, the cells. It works beautifully in cells, but if you inject it in a human, there's going to be a lot of side effects, and so you cannot really use that to cure a person. So there's many reasons why clinical trials fail, but I think that one of them might be where people have altered the results to their advantage in the earlier stages, the preclinical work.
And so much of that is statistical in nature, in addition to image manipulation. And so I wonder, do you have aspirations of building out a more permanent team where you have multiple different subject experts that are able to look holistically at a paper as opposed to just being limited to images?
Because there's also this whole other level, which we haven't even talked about, which is that interpretive stage of where you have the data, and maybe the data is pristine, but you can, portray it with the x-axes and jumps in the different axes, or you can, highlight some data versus other. Like there's a lot of ways I can imagine that one would manipulate something to whatever incentive is driving them. And it actually gets really sketchy when you think about pharmaceutical industries analyzing the results of these clinical trials and putting out the papers, essentially, because it's like, well, you have a clear reason to make something look good. I wonder if there's like a systematic way, like bouncing off what you're saying, Anastasia, how you would go about analyzing those kind of problems in a paper down the road, maybe.
Yeah, you're right. There's tons of different ways you can look at a paper and find different types of problems depending on what type of proverbial glasses you put on and look at the paper from different angles. So I'm not good in statistics. I will I recognize my strengths and my weaknesses. That's not something I will ever be able to do well, and I'm already completely swamped with requests to look at images, so I'm not going to be out of things to look at anytime soon. But other people do. We don't really work in an organized team. We're sort of loosely organized sleuths who all look at papers from different angles, but other people look at statistical types of problems or citations or interpretation errors. And of course, you can, if you apply multiple statistical tests and you only choose the one that gives you some significant outcome, otherwise called p-hacking, you can perhaps get away with it, but people who know much more about statistics than I do will sometimes find those problems in papers and post them also on Pubpeer. So there are people doing that. It's just not me, because I can only find very, very simple things like, these groups do not look significant with the eyeball test, where you just look at the graphs and you're like, how can this possibly be statistically significant, because they look exactly the same to me. And so I might raise that, but when it's a little bit more complex than that, that's not me who can raise those concerns.
Do you have aspirations to, I hate to say, to institutionalize what you do, but to make it more, to have a clear path of succession? Because at some point, that's what all projects run into, right? You have somebody who's kind of a visionary, who sees things in a different way, in your case, literally. Mm-hmm. And as you focus on it less with time, is there a way for you to train people to come up behind you who can do the same thing? Is that something you're interested in? Or are you just sort of focused on the details of individual papers?
Yeah, I try to train people by posting my concerns on Pubpeer and also on Twitter. So when I post on Twitter an image from a scientific paper without my boxes highlighting the duplications, I turn it into a game. So I try to teach people how to do it in a hopefully playful and fun way, because people, the first three who give me the right answer, will win an emoji award. So like people are on it, like I want to win the award, and I'm too late, and they're very excited about playing the game. But yeah, the idea behind that obviously is, when people look at papers as a peer reviewer or a reader, to look at them in a slightly different way. Give the paper a couple more seconds or minutes to look at the images and spot these problems automatically. So I think once you're pointed out these issues, these problems, these duplications, you might spot them yourself. And not everybody can do it, because I would guess about half of the people can also see these problems easily once they know what to look for. The other half is just not able to ever see it. Like, I guess, some people can whistle and some people can sing and others cannot. So if you have that talent, you might spot these problems. So I have a lot of followers on Twitter, so I hope to have educated a lot of people in these things, and they're starting to send them to me like, I found one myself. So good. So in that way, I'm trying to train other people to do that. Also to inspire other people to speak up when they see something wrong, either in their own lab or in other labs. I'm not really planning to start an official team. One thing I've learned in my previous jobs is that I really, really hate meetings, and I just want to do my work, and just tell me what to do in 5 minutes. I don't need a whole hour of a meeting to do so. Just send me a list of things to do. I'm in, but I don't want to sit in these very long meetings. So I feel very unproductive during that time. So if I would run an institution that trains all these people, I would, I'm sure, be sucked into meetings, and I would not have time to do what I do best, which is looking at papers and images. So I want to not go into that at all. I just want to focus on what I do, and I'll give talks and workshops and things like that, but I do not want to be an official leader or manager of a team.
Yeah, the only thing I miss about meetings is the free lunch.
Oh, yeah, well.
Lots of good memories from
you don't get that over Zoom.
Nowadays are on Zoom, right? And there's just no free lunch.
Absolutely not. Absolutely not.
No free
I guess
bagels.
My maybe to wind this down, what do you see yourself as starting a branch of law enforcement in science? Are you like the science sheriff? Like how do you view yourself?
I think no, I'm not the sheriff, because I cannot really investigate these cases. I can only look at publications and see which images have been published there. But I don't know the behind the scenes things, like how did the real image look like? Who did which experiment? Who's responsible? For that, you need to have access to lab books and emails and
science detectives.
Yeah, so I see myself as a detective, for sure, because I do follow leads like, oh, this paper has a problem, and who are the authors on it? And I don't know who it was, but let's follow all other papers from the first author. Oh, there's no problems in them. No, let's look at papers from the last author, who is usually the senior author. And so I might follow leads like that. But then, yeah, I cannot really do the follow-up research. So I don't see myself as a sheriff, although sometimes I've voiced my frustration about the lack of reply or the lack of action from journals or institutions investigating these things. So I have opinions for sure, but I cannot force them to investigate or look at particular things. They do their own thing, and I don't have any influence on that.
Well, I think we've brainstormed at least a few steps in the right direction of positive incentives, and I think this Pubpeer is encouraging, right?
I love Pubpeer so much. It's so gratifying to be able to take the DOI of a paper or a title and to throw it in there and to see people are talking about it. And
right.
And it might not result in capital punishment for these PIs.
You're just so obsessed with this idea.
I mean, they might not get canned right immediately, but the more eyes that are on these problems, I feel like it does put a pressure on those people to behave.
Yes. And institutions to take action, because we have seen very little action from either publishers in retracting these papers, with some exceptions, obviously, or institutions to investigate. It's almost like we have traffic without the chance of getting a speeding ticket. Or it's like some sports category where doping is fine, where we all look the other way and we don't really punish people who are caught doing doping and win the race. And I think in both in traffic as well as in sports, there has been the realization, like, we need to have rules and we need to have some enforcement of those rules, because otherwise traffic will become a chaos, or the person with the most injectable drugs will win the bike race or so. Like, in those fields, we have realized we need to have enforcement and rules. In science, not so much. I mean, there's all beautiful guidelines, but there's just very few cases where the Stanford president, I think, was an exception, where there was actually a person stepping down. But the university wanted to investigate this internally. They wanted to keep it under wraps, and it was only because of a lot of people voicing their displeasement with that that they then started an outside investigation. But they wanted to keep it a secret, and now the outcome is that the president stepped down. But it's very rare to see that.
I think that's a really profoundly wide-reaching point too about guidelines in general, is that if you don't have consequences for breaking a guideline, what is it? It's just sort of this half-hearted hope that everybody will like the guideline and go along with it.
Right.
I think rules have to be enforced, as much as it's, it seems brutal. Like, nobody really likes the police when they get pulled over, maybe, but it's, in some sense, you're also just like, well, then why even have speed limits if we're not going to police them? So I can see the value in that for sure.
And it makes all of us a better driver. If I see a car being pulled over, I'm like, oh yeah, I shouldn't be speeding, right? I mean, it helps.
I should stop going 90 in this 45.
Because it is very, if we speed, it's rewarding, right? Because we'll be at our where we need to be. We will be there.
It's fun too. It's fun.
Fun, yes, but no, there need to be rules and there needs to be some enforcement. Otherwise, it will be chaos.
Agreed. Well, I know you have to run. I hope you guys have a wonderful trip down to Southern California today.
Yes.
I kind of wish I was getting out of this rain and down to Southern California as well.
Well, the rain is actually on its way to Southern California, so we're definitely going to hit a lot of rain later on this week. Yes.
Well, that'll be good for the desert.
If you have, I don't know how much time you have, and I know that Death Valley's a little bit of a trek, but apparently there's this lake that just formed in Death Valley from all of the rains, which is this liminal lake that only forms, every like 50 years or something.
That's not going to be on our route this time, but I've been to Death Valley many times, and it's a wonderful place, and it changes with the seasons. If there's rain, I really look forward one day to go there when we have these super blooms and all the things that make the desert look very different on that day.
Yeah.
I've only been there once, and our car's air conditioner broke, and
Oh,
so
no.
it was just
That
like
is-
having a hairdryer in your face inside of a
that is
sauna.
bad. Yes.
Oh, it's
Yeah. No,
unbelievable.
it can be a dangerous place if things break down, for sure.
I hope that we get a chance to talk to you again, because I think that there's a larger philosophical question here of science and truth and what it means to come up with something that you can actually rely on and
Mhm.
use without having to consistently be reevaluating it. And that's kind of outside this conversation, but I also think that it's a really important conversation for science, because that's what we have to orient towards. That's the positive reward.
Right.
The capital punishment is the disincentive, but there's also something very beautiful there.
Yeah, there is, and I think it's hard for people who have never done science themselves to understand how we will probably never reach the truth, whatever that is. But it's a path to the truth, and it's a winding path. It goes to the left, it goes to the right, it finds this, it finds that, and it contradicts itself, and it's complicated, and it's beautiful if you are a scientist yourself. But I think if you don't have that background, it's really hard to understand how long, for example, it took to figure out if the coronavirus was aerosolic, or how did it transmit, and does wearing a mask help or not. So there's just so much conflicting data, especially that came out in the early days of the pandemic. But that's how science is. It's not going to be a straight answer from day one. It's going to take a couple of years to figure out these things. And I think it's hard to understand how slow and how difficult science can be to reach a particular answer. And then maybe 10 years from now, we might reach a different conclusion because there's new insights. And so science is always changing. There might not just be a truth because we don't have enough data to know what the truth is. We will never have enough data. But we will eventually get close to the truth. But it's going to be a very complex. It's not like A equals B. It's going to be, a very complicated formula that we now have all the parts to fill out. But we'll never reach that truth, I think.
Yeah.
Amen.
Yeah.
Yeah.
I hope we get a chance to talk about it. I hope you come back again. It was really wonderful. Thank you.
I'd love to, yes. Well, thank you for having me on. I enjoyed talking to you from my walk-in closet. And wishing you both a wonderful day.
Thank you.
You as well.
We salute
Thank
your
you.
efforts.
All right.
Have a great
Bye-bye.
trip.
Bye-bye.
Bye, everybody. See ya.